All right, in the last lesson we looked at water and how water gets in and out of cells. Now what we're looking at in this next lesson is how other things, so this is about big items, like salts and sugars, get in and out of cells. So now we're not talking about simple osmosis and water being able to sneak across a membrane where it could sneak past shifting phospholipids. Now we're talking about big things. This is a salt or a sugar molecule or even a small protein that might like to get in. And there's two ways that big things get into cells. And both of these ways require a protein. So these are examples of proteins that are facilitating diffusion. So this is facilitated diffusion. Now in this case, these molecules are moving past a membrane, but they're not moving through the membrane. They're moving through a protein instead. So that's why this is not called osmosis. This is known as facilitated diffusion. And it's a type of passive transport, okay, because things are moving with the concentration gradient, okay? So this is still passive because you're moving with this concentration gradient. And you'll remember from the last video that to move with a concentration gradient means you're moving from high concentration to low concentration. And again, passive is going to mean that there's no energy involved then because it's just moving with the concentration gradient. So the first example is the channel protein, and that's what you're seeing here. The channel protein is what this picture is showing you. This is a protein that's kind of like a straw stuck into the plasma membrane. It's this little short, stout protein that's got a hole that runs through it. And that hole is big enough for some solutes to pass. So that's what's happening with a channel protein. That one is really easy to understand. Okay, so this is literally just a hole in a protein. Okay, that's literally what it is. Okay, now in the second picture that you're looking at, our protein is going to be called a carrier protein. Okay, and what I'd like for you to do is kind of divide this picture in half like this. This is the before shot. This is the after. Okay, so before and after. Carrier proteins actually change shape to allow things to go through. So the channel proteins just have this shape that easily lets stuff go through. Carrier proteins are shaped like this, and then they change their shape to allow stuff to go through. You can think of this kind of like the swinging doors on a saloon in a Western. It doesn't take a whole lot of energy to get those doors to swing. It doesn't really take much energy at all. If you just start walking and bump into them, they will swing. Okay, with no effort. So this still requires no energy because you are moving with a concentration gradient. So the protein looks like this. As the molecule bumps into it, the protein changes shape and allows it to get in. So these are both examples of facilitated diffusion, and they're using a protein to accomplish that. Now, there's another way that big stuff gets in, and that other way is through something called active transport. Now, active transport through a protein happens because you are moving against a concentration gradient. In other words, we are going from low concentration to high. Okay, so low to high. you'll notice this looks the same as our carrier protein before. Okay, so our molecule still bumps into the protein. Unfortunately, this molecule is moving backwards. There's a low concentration of these molecules outside of the cell, and there's a high concentration on the other side of this membrane. So when this molecule bumps into the protein, it doesn't automatically change its shape. It's going to require some energy, and for now, that energy is ATP. So for now, just know that ATP is energy. Next unit we'll talk more about ATP, but every time you see ATP, read that as energy. Okay, so in this case now, we're trying to stockpile molecules on one side of a membrane. Okay, so like in here, and we have very few of those molecules outside, so we're having to use energy and force the molecule to go through that protein, and that energy is in the form of ATP. Okay, so against the concentration gradient, low to high concentration, and we're using energy. All right, let's just sum up transport and what we see about transport now. Okay, transport across membranes can either be passive or active. Okay, now all the passive forms of transport use no energy. And that's because you're going with a concentration gradient, which means from high, from high to low concentration. So all the passive transports have no energy because they're going from high to low concentration. There are several forms of this. There's diffusion, okay? If we have diffusion across a membrane, that's osmosis. A type of diffusion really is facilitated diffusion. And in facilitated diffusion, you're not going through the membrane. Remember, you're using a protein, okay, to get in. And facilitated diffusion is for big stuff. Osmosis is for water, okay? Now, there's also active transport. And active transport uses energy. And the reason why it's got to use energy is we're going against the concentration gradient, which means we're going from low concentration to high. Okay. The one that you just saw involves the protein pumps. And by the way, when you're taking that carrier protein and you're going against the concentration gradient, we don't call that a carrier anymore. We call it a protein pump. There's also another form of active transport, endocytosis and exocytosis. And in endocytosis, this looks a lot like what you saw with endosymbiosis, where a cell kind of reaches out and tries to grab a food particle and pinches it off into a vesicle like this. This is the endocytosis version. So there's this little food particle right here, and the cell is kind of reaching out, grabbing it, and pinching it off inside. That's bringing in things that are even bigger than salts and sugars. Exocytosis would be if we reverse this process. So the red arrows would be exocytosis. If we had something that was inside of the cell here, and say now this food has been digested and all that's left in this little pocket is waste. If that little pocket goes and fuses with the membrane to dump that waste out, that's exocytosis. So endocytosis would be the black arrows, right? Okay, black arrows. Exocytosis is the red arrows. Whoops, there we go. Okay, so those are types of transport. So that kind of gives you an idea of all the different types of transport there. All right, finally, why is this so important? Obviously, transport is important to individual cells, but remember that transport is responsible for the process of absorption and releasing waste, right? So you're absorbing things you need and you're releasing the waste. More membrane equals more absorption and waste release, right? So the more membrane that you can fit into a small space, the better, right? So for example, if I take two lines here, here's a line and here's a line. which line is bigger, the top or the bottom line? Well, obviously, this bottom line is bigger, right? I've been able to get more line in the same amount of space by folding my line, right? Well, it's the same thing with membranes. You can have straight across membranes, right? And those will do a certain amount of absorption and waste removal. But if you can fold your membrane up, you can even get more absorption and waste removal. The reason why this is important is there will be times where, say, you could have a question on the EOC that says, which of these pictures would look the most like the inside of your intestine? And it would give you something like this, right? Well, if you remember that the intestines are for waste removal and absorption, they're not going to be smooth, right? They're going to be foldy membranes on the inside of your intestine, right? Because the intestines are doing a bunch of absorbing and then also waste removal. So every time you see folds or folded membranes or folded organs, understand that that's all about absorption and waste removal. So you're kind of increasing the surface area for accomplishing those two tasks in the same amount of space. All right, well, that's it for transport. You've now covered everything in this unit on cells.